Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, let's dive in. Acetaminophen, stuff in Tylenol and countless other products, it's one of the most common pain relievers out there. First made way, way back in 1877.
Yeah, incredibly common. Almost everyone has it in their medicine cabinet.
Exactly. But here's the kicker. We still don't fully understand exactly how it works, how it actually stops pain and fever. It's kind of wild, right?
It really is. For something used so widely for so long, it's surprising the mechanism isn't completely nailed down.
And it's not just a curiosity thing either. This drug, as common as it is linked to roughly 500 deaths every year in the U.S. alone, mostly due to liver damage.
That's the really serious side of it. High doses can be quite dangerous, which makes understanding how it works even more critical.
So that's the puzzle we're looking at today. How can this thing we all use, which carries real risks, still be such a mystery in terms of its fundamental action?
It really shows how complex pharmacology can be, even with drugs we think are simple.
Absolutely. And that leads us to our mission for this deep dive. We're going to unpack some really fresh research just published in Cell Reports Medicine. It's from scientists at Indiana University.
Right, and they've potentially found a new piece of the puzzle, a new way acetaminophen might be relieving pain.
Yeah, so we'll be digging into that paper and also a news piece from the university that helps explain it. The goal is to get our heads around this potential new mechanism. Understand why it's kind of surprising and what it could mean down the road for pain treatment.
And we'll try to keep the science clear, avoid getting too bogged down in jargon.
Definitely. So before we get to the new stuff, maybe a quick recap on what scientists previously thought. What were the older ideas about how acetaminophen works?
Well, one main theory involved enzymes called cyclooxygenase, or COX enzymes, especially COX2. The idea was that acetaminophen might inhibit these, particularly in the central nervous system.
Right, and those enzymes are involved in making prostaglandins, which signal pain and inflammation.
Exactly. But the catch is that acetaminophen isn't a very potent inhibitor of these enzymes compared to drugs like ibuprofen. You often need quite high concentrations to see that effect clearly.
Okay, so maybe not the whole story. What about the cannabinoid connection? I remember hearing about that.
Yes, that's another hypothesis. The body can convert acetaminophen into a metabolite called N-arachidonolambda-phenol. Let's just call it AM404 for simplicity.
AM404, got it.
And this AM404 molecule was thought to interact with the endocannabinoid system. That's the body's own cannabinoid signaling network. Maybe by boosting levels of an endocannabinoid called anandamide, for example.
Endocannabinoids being the body's natural cannabis-like molecule.
Precisely. But again, there were questions. The levels of AM404 actually produced in the body after taking acetaminophen seemed, well, maybe too low to fully explain the pain relief through that road alone.
So holes in the existing theories, which makes this new research really timely.
It does. This new study also looks at the endocannabinoid system, but from a completely different angle.
Okay, tell us about it.
So the researchers focused on a specific enzyme. It's called diacylglycerol lipase alpha. That's DIACYGLYCROLLIPASE alpha.
Diacylglycerol lipase alpha.
Okay, and this enzyme is really important because it produces one of the main endocannabinoids in our bodies called 2-arachidonolglycerol. Let's spell that out too. 2-arachidonolglycerol. People often shorten it to 2-AG.
Got it. Diacylglycerol lipase alpha makes 2-arachidonolglycerol. So what did they find acetaminophen does to this enzyme?
This is the core finding, and it's really unexpected. They discovered that acetaminophen actually inhibits diacylglycerol lipase alpha.
Yeah.
It slows it down.
It inhibits it, so it reduces the production of that endocannabinoid 2-arachidonolglycerol.
Exactly. Less enzyme activity means less 2-AG gets made.
Huh. That seems backwards, because isn't the general idea that more endocannabinoids mean less pain? Like, that's the whole principle behind medical cannabis for pain, isn't it?
That has been the prevailing view, yes. Increase endocannabinoid signaling, decrease pain. But this study flips that script, at least for 2-AG in this specific context. It suggests that reducing 2-AG levels might actually be part of how acetaminophen relieves pain.
Wow, okay. That is genuinely surprising. So how did they figure this out? What kind of experiments did they run?
They used a multi-pronged approach. First, they did in vitro work studies in lab settings. They used a special setup with single nerve cells called autoptic hippocampal neurons.
Okay, what does that let them see?
It's a neat system where a single neuron connects back to itself. It allows researchers to study cannabinoid signaling in a very controlled way. And in these neurons, they found acetaminophen inhibited a type of signaling that relies on cannabinoid receptors, specifically something called depolarization-induced suppression of excitation.
So basically, in these isolated nerve cells, acetaminophen messed with a cannabinoid-related communication pathway.
That's the gist, yes. It suggested a direct effect on cannabinoid signaling at the neuronal level.
Okay, what was next? Did they look directly at the enzyme?
They did a couple of things. They used engineered kidney cells, human embryonic kidney cells, that were made to produce lots of that enzyme, diacylglycerol lipase alpha.
Right.
When they stimulated these cells to make 2-arachidonyl-glycerol, adding acetaminophen resulted in lower levels of 2-AG being produced. That directly supported the idea that acetaminophen was hitting the enzyme responsible for making it.
So confirming the enzyme link in a cell system.
And they also did biochemical tests, directly measuring the enzyme's activity. They found acetaminophen reduced the activity of diacylglycerol lipase alpha, but interestingly, not a related enzyme called diacylglycerol lipase beta.
Alpha, but not beta, so it seems pretty specific.
That selectivity is potentially quite important, yeah.
Yeah.
It suggests a specific target, not just a general effect.
They also used some kind of sensor, didn't they?
Yes, they used cells equipped with a biological sensor that lights up essentially when 2-arachidonylglycerol is present.
Yeah.
And again, when they stimulated 2-AG production, acetaminophen reduced the signal from the sensor. So another way of showing less 2-AG was being made.
Okay, so multiple lines of evidence from cells and biochemical tests, all pointing to acetaminophen inhibiting diacylglycerol lipase alpha and reducing 2-AG. But does this actually relate to pain relief in a living animal?
That's the crucial next step, the in vivo studies. They use mice and a standard pain test called the hot plate test.
How does that work?
You place a mouse on a warm surface, not dangerously hot, but uncomfortable, and measure how long it takes for the mouse to react, like licking its paws or jumping. A longer reaction time suggests pain relief.
And what happened with acetaminophen?
Acetaminophen increased the reaction time, meaning the mice showed signs of pain relief, as you'd expect.
Okay, standard effect. But did they link it to the cannabinoid system?
Yes, and this is key. They tested mice that they were genetically engineered to lack cannabinoid 1 receptors. These are the main receptors that 2-arachidonylglycerol acts upon.
Right.
In those mice, acetaminophen didn't provide pain relief on the hot plate test.
Ah, so the pain relief seems to require those cannabinoid 1 receptors to be present and working.
Exactly.
Yeah.
It strongly suggests that pathway is involved.
Ah.
And they did one more thing. They used a different chemical, not acetaminophen, that also inhibits diacylglycerol lipase.
Okay.
That inhibitor also provided pain relief in normal mice. But again, it didn't work in the mice lacking the cannabinoid 1 receptors.
Wow. So inhibiting that specific enzyme, whether with acetaminophen or something else, seems to reduce pain through the cannabinoid 1 receptor pathway.
That's the conclusion they drew from the animal studies, yes.
So let's try to summarize the proposed mechanism based on all this. How is acetaminophen working according to this new research?
Okay, so the idea is acetaminophen gets into the relevant parts of the nervous system. It inhibits the enzyme diacylglycerol lipase alpha. This causes less 2-arachidonylglycerol to be produced.
Right, lower levels of 2-AG.
And because there's less 2-AG floating around, there's less activation of cannabinoid 1 receptors in certain pain-related circuits. And somehow this reduced signaling through CB1 receptors leads to pain relief.
It's fascinating because it's so indirect and counterintuitive based on the old more endocannabinoids equals less pain idea.
Absolutely. It really challenges us to think more nuancedly about the endocannabinoid system.
So what are the big takeaways here? What are the implications of finding this potential new mechanism?
Well, first, it just gives us a fundamentally new perspective on how this extremely common drug might be working. It adds a whole new layer involving the endocannabinoid system, specifically by reducing a key endocannabinoid.
Which could explain some of the effects that the older theories maybe didn't fully capture.
Possibly, yes. It might help reconcile some inconsistencies.
And what about the practical side? Drug development. You mentioned the liver toxicity issue earlier.
That's a huge potential implication. If diacylglycerol lipase alpha is indeed a key target for the pain-relieving effects, maybe we could design new drugs that specifically target this enzyme.
Ah, so you could aim for the pain relief without hitting whatever causes the liver problems.
That's the hope. If the liver toxicity comes from other actions of acetaminophen, a drug that only inhibits diacylglycerol lipase alpha might be effective for pain, but much safer. That's what the researchers themselves suggest as a possibility.
That would be a massive win for pain management.
It certainly would. And beyond acetaminophen, it also makes us rethink the role of endocannabinoids in pain more generally.
How so?
Well, finding that reducing 2-AG can relieve pain suggests the system is more complex than just activate receptors, feel better. Maybe different endocannabinoids or the same ones in different circuits have different roles. Perhaps 2-AG in some pathways actually promotes certain types of pain signaling, and reducing it is beneficial.
So it adds a lot more complexity to the picture.
But definitely, more nuance.
Now, like all research, this study must have its limitations, right? Things it didn't fully answer.
Oh, absolutely. Good science always acknowledges limitations. One big one is they haven't pinpointed exactly where in the brain or nervous system this diacylglycerol lipase alpha inhibition and subsequent CV1 effect is most critical for pain relief.
So they know the enzyme and receptor, but not precisely the where.
Right. Mapping those specific neuronal circuits is a next step. Another point is that the in vitro work focused mainly on neurons.
Just the nerve cells.
Yeah. But we know other brain cells like glial cells and microglia are also involved in pain and cannabinoid signaling. This study didn't fully explore their potential role in this mechanism.
Okay, so other cells might be involved too. And what about the types of pain? They use the hot plate test.
Which is good for measuring acute thermal pain, that quick response to heat. But we don't know yet if inhibiting this enzyme would work for other kinds of pain, like chronic pain, nerve pain, inflammatory pain.
Right. Different pain types might involve different mechanism.
Exactly. So we need to see if this applies more broadly. And probably the biggest limitation, or rather the next big question,
is whether this all translates to humans.
Precisely. These are fantastic findings in cell models in mice, but we need research to confirm that inhibiting diacylglycerol lipase alpha is actually relevant to how acetaminophen relieves pain in people. Animal models don't always predict human responses perfectly.
That's always the crucial step. Now, you mentioned how surprising this finding was. Let's bring in some quotes from the researchers. Mikhaila Dvorakova, one of the lead scientists, said something quite striking.
Yes, she said. Up until now, we thought that elevated endocannabinoids in our body meant less pain, but our study shows that in the case of 2-arachidonyl-glycerol, it might be the opposite. Actually, reduced levels of 2-AG leads to decreased pain.
That really captures the core surprise, doesn't it? Turning that previous assumption on its head.
It does. And then Alex Straker, another key researcher, commented on the drug development angle.
What did he say?
He said, if you don't know what the target is, you can't design an alternative.
Sure.
Our research suggests that this enzyme might be the target, in which case you can start developing drugs that target that specific enzyme, but without that toxicity.
That highlights the potential path forward, the practical benefit of identifying this enzyme.
Exactly. It provides a concrete target for future drug design efforts.
And it's worth noting, apparently there was some skepticism initially in the scientific community when these ideas were first presented.
That's often the case with findings that challenge established models. The news article mentioned that, yes, given the long-standing idea that activating cannabinoid receptors is generally analgesic, proposing that reducing an endocannabinoid could also relieve pain definitely raised some eyebrows.
It just shows how science progresses, right? Challenging ideas, testing them rigorously.
Precisely. It needs solid evidence, like what this paper provides, to shift perspectives.
So as we wrap up this deep dive, what's a final thought for our listeners to take away from this?
I think the main thing is that our understanding, even of very common medicines, is constantly evolving. This research is a great example. It opens a new window onto how acetaminophen might work and potentially offers hope for developing safer, more targeted pain therapies by focusing on this diacylglycerol lipase alpha enzyme.
It's a reminder that there are still mysteries to solve, even in our own medicine cabinets. And for anyone interested in digging deeper, the paper is in Cell Reports Medicine, published in 2025, or just exploring research on the endocannabinoid system in pain. It's a really active field.
Absolutely. And it's ultimately optimistic, I think. It shows the dedication to understanding pain better and finding improved ways to treat it.
So just to quickly recap the core idea one last time, this new research suggests acetaminophen might relieve pain, at least partly, by inhibiting an enzyme called diacylglycerol lipase alpha.
Which leads to lower levels of the endocannabinoid 2-arachidonyl-glycerol.
And surprisingly, these lower levels, acting through cannabinoid 1 receptors, seem to contribute to pain relief in the models they tested.
Exactly. Really fascinating, somewhat counterintuitive mechanism.
Well, thank you for walking us through that complex but really interesting research. We hope you, our listeners, found this deep dive insightful.
Thanks for tuning in to the Health Headliner podcast by To Do Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.